we'll go through the rest of it, and I'm going to turn the fire hydrant mode on. So we'll just go over the review, and you guys just write. Obviously, you can stop me and ask a question, but I'm just going to go through it. All right, so just starting from the beginning, we already looked at what is science. It is a process of discovery, discipline of testing, of inquiry, testing, and inference. What is biology? The study of living things. Know a few examples of how it impacts our world. So again, that's the why biology part of it. What is my philosophy of science? That's the whole curiosity leads to fascination, leads to questions, experiments, knowledge, and you can even add worship at the end, because as believers, the more we know about the world around us, the more we should be in awe of the one who created and designed it. How can I apply this to my career or dream job? That's just up to you guys. I'll put that on there, but that's just creativity. Okay, 1.1, the process of science, scientific method. The general gist is observation leads to question, leads to a hypothesis, leads to testing. You can put prediction in between the two if you want, but testing and then analyzing the results, right? So just be familiar with the flow of it. Practice applying it, like the flashlight example. Just know how you actually do this all the time, whether you know it or not. What is a scientific theory? We saw that it is not, my theory is. It's actually a hypothesis that has been used to make successful predictions time and time and time again for a long period of time. Right, so there's a lot of evidence to suggest that that is probably what's going on, more than a hypothesis. 1.2, what is the difference between independent and dependent variables? Independent is the variable that you are changing. Dependent is a variable that is dependent on it. So, for example, we said a silly example, the Starbucks coffee scientists. The independent variable was the type of bean they were using. It was just the one thing they were changing. Everything else was the same, right? So, and then the dependent variable was what they were measuring, the caffeine. So that's dependent on the other variable. What is a placebo? That is a ineffective treatment. So something you know won't work, basically, that the subject doesn't realize is different from the real stuff. And that's just used as a control. What is the problem with secondary sources? They're biased. There's a slide on it. You can read more about it. But, it's someone's interpretation of the primary source. So it's going to be a little biased. They might not have the whole story. It is someone's interpretation. So they'll only list a part of it. Whatever. What kind of sources are the most reliable? Primary sources. That was to answer your question, Emily. Okay, 2.1. Know what atoms and molecules are. Know the different parts of an atom. Protons, neutrons, and electrons. Then this is the thing that you should be starting to say in your sleep. What four organic molecules do all living things have in common? Nucleic acids, proteins, lipids, and carbohydrates. Why is water important? Just the different ways that we saw that water can sustain life. The most important one, in my opinion, is that it participates in chemical reactions. So without it, we couldn't do photosynthesis, we couldn't do cellular respiration. But obviously there's a lot of reasons. What gives water its unique properties? Firstly, the fact that it's polar, and that allows it to form hydrogen bonds. And you should know what polarity, hydrogen bonds, hydrophilic, hydrophobic, and electronegativity mean. Electronegativity, just to remind you, is how electron hungry an atom is, right? How greedy it is for electrons. So oxygen is very electronegative, so it pulls electrons toward it. And that's what gives water its polarity, actually, is because oxygen pulls electrons toward it, so it becomes slightly negative. Know what cohesiveness means and what molecular bond causes it. Again, hydrogen bonds. In this test, when in doubt, and you see hydrogen bonds as one of the options, you can circle hydrogen bonds, and you'll be fine. What are organic molecules? We just looked at that. Molecules that have carbon and hydrogen. What are hydrogen bonds? We looked at that. Slightly positive charge on a hydrogen interacting with a negative charge on any atom. Oxygen, chloride, etc., etc. Are they the same as the bonds between hydrogen and oxygen within a water molecule? No. The hydrogen and oxygen that form a water molecule are strongly bonded. That's a covalent bond. All right, the next under structure and function of macromolecules. What organic molecules all living things have in common? Again, the 4 that we've looked at. What are polymers and monomers? We went over that. What is the name of the reaction that creates polymers from monomers? Dehydration synthesis. What is the name of the reaction that creates monomers from polymers? So the one that breaks them apart, and that's hydrolysis. Lysis means to break apart. And the why is it called that is simply just for you to know that the name means something. So know that dehydration is removing a water, synthesis is bringing it together, right? Why are carbs important? You guys got that right, because they provide most of the energy in our body. They're a quick energy. The monomers are monosaccharides, polymers, you can say polysaccharides or carbohydrates. What is sucrose made from? You don't have to memorize that. I'm not going to ask that on the test, but it's good to know. Okay, it's fructose and glucose, 2 monomers that come together. And what is high fructose corn syrup? Now you know it's just a little bit more fructose than glucose in the mixture. And that's the only difference, is 5% of one monomer. And people freak out about it. Nucleic acids. Why are they important? They contain the genetic information. What are the 2 types, RNA and DNA? How do their functions differ? There, you should be able to answer that based on the central dogma, right? What is the main difference between DNA and RNA? The main difference is that one is double-stranded, one is single-stranded. There are obviously other differences. One has one uses thymine and the other one uses uracil. That's a difference. One has ribose as its the sugar in the nucleotide. The other one has deoxyribose. But the main one is the strands. I even put it there in parentheses just to throw you a soft one. What are monomers called? Nucleotides. Why is DNA called a double helix? Again, 2 strands that twist into a helix, right? Know what kind of interactions allow the 2 strands of DNA to interact to form a double helix. And those are hydrogen bonds. Yeah, awesome. Proteins. Why are proteins important? They are the workers of the cell. What are the monomers of proteins called? Amino acids. What are enzymes? Know a few examples. So enzymes are proteins that I'm trying to remember the definition I gave you, but that speed up a chemical reaction. Yeah, enzymes are a protein that speed up a chemical reaction. And actually, I guess I didn't put that, but the way they do it is by lowering the activation energy. I guess I didn't put that because that's a little bit too detailed. And then the few examples we'll see in a bit. So what are 4 layers of protein structure? Primary, secondary, tertiary, quaternary. Just look over that. But know that the primary is the one that determines the shape, right? The overall shape is determined by the sequence. And what helps amino acids interact to form a secondary structure? Hydrogen bonds. Again, you'll see that was the last time you will see hydrogen bonds. They now make their exit and leave the stage. We're left with no more. Let's see. Ooh, this will not fit. It's okay. I think it'll last. Okay. Lipids. Why are fats important? We talked about that. Energy storage. Long-term energy storage. Are lipids polymers? No. And this is actually a tie-in to our cell chapter. And I'll ask you guys, what part of the cell is made up of lipids? Is mainly made up of lipids? The plasma membrane or the cell membrane. And someone else, why is that? What's the molecule that they're made up of? Who remembers? They're like, it is like you tore off the arms off a stick figure and its torso, and now it's just the head and 2 legs. You know it?
Yeah.
Phospholipid, right? And that's easy. Phospholipid. So lipid is the molecule. Are lipids, obviously. Lipids are lipids. So phospholipid is the one that makes up most of the cell. So it's really important. All right, so 2.2, prokaryotic and eukaryotic, those are the 2 cell types. The differences, main one is a prokaryotic does not have a nucleus. Cell structure, what are the cell membranes mainly made of? Phospholipids, right? Those are also an important one. I highlighted there, I bolded cell structure. You should spend time looking at that. What is the function of the plasma membrane? Basically, to keep things that the cell wants inside, and the things that it doesn't want inside, outside. And the concept that goes with that is selectively permeable, right? It's selectively permeable. So it lets water come in and out, but it doesn't just let anything come in and out. Why is it called a fluid mosaic? Because mosaic first because it's not just phospholipids, there's proteins and carbohydrates, and they all move around, which is why it's called fluid. Why are the mitochondria important? They generate energy. Where does photosynthesis occur? We looked at that when looking at plant cells and chloroplasts in plant cells. What do I compare a cell to? A factory. And there you should go through the slide and see how the different organelles compare to different parts of the factory. And that'll help you. It'll help you understand what a cell is made of. All right, last chapter, 9.1. What is the central dogma of biology? It is how genetic information flows from DNA to RNA to protein. And how is it like making a meal from a recipe book? Just understand the analogy there and be comfortable with it.
You said DNA to RNA?
DNA, RNA, yep. And DNA to RNA. Just always remember DNA is the master molecule. It is the one that steals the show in biology, right? So it's always the first. All the information is contained in DNA. What enzyme is used for transcription? So this is back to the enzyme part. Notice a few examples of enzymes. What enzyme is used for transcription? Anyone remember? RNA. And then think of
DNA?
No, it's RNA is the first part of it. But then think of what you're making. So you're taking little building blocks and putting them together. What is that called? What is it when you put monomers together? Polymer. So RNA polymer ACE. So RNA polymerase. Anytime you have an enzyme, it'll end with ACE, except for a few like ribosomes. So that is the enzyme, is RNA polymerase. And it's right there below it. How does RNA polymerase know where a gene starts? It's promoter, right? There is a slide that kind of illustrates all of that. What is the name of an RNA copy of a gene? An mRNA, messenger RNA. Why does mRNA have to be exported out of the nucleus? We saw that in the last slide that we looked at, but it's because it has to load onto ribosomes so it can be used to build a protein, right? Otherwise, if it stays in the nucleus, there are no ribosomes in the nucleus, so it would never be used to make protein. All right, translation. What enzyme is used for translation? Ribosomes. Right on. What are they made of? Protein, RNA, or both? They're made of both. Yes, both. How does the code in mRNA specify amino acids? So there you should just know that the code in an mRNA has codons, which code specifically for an amino acid, which code for a specific amino acid. What is a codon? A 3-letter sequence in the mRNA that specifies an amino acid. This is kind of repetitive stuff, but you should know it. How are tRNAs paired up with their respective codons? By the anticodon, right? The sequence at the end of it that matches the sequence on the mRNA. And then how does the ribosome know how to add tRNAs in the right sequence? And that's just simply by the codon sequence, the sequence on the mRNA. So it's a little bit repetitive stuff, but it's because if you understand the concept, then you know all these questions. You don't even have to memorize. That's the beautiful thing about understanding a concept. You understand it, and then you can look at these questions and be like, I didn't even have to memorize the answers because I know how it works, you know? All right, last two. In what molecules do mutations initially occur? DNA, RNA, or protein? I'll ask you guys that. DNA? RNA, someone said?
It usually occurs in the transcription process.
It can occur in the transcription process, but DNA is the right one. So it actually will randomly occur in the replication process, which we haven't looked at yet, but it's before a cell divides, it replicates its DNA. It doubles it. So in that process, sometimes it can introduce a mutation. But DNA, right, that's where mutations occur. So that change of sequence in the DNA affects the sequence in the protein because it affects the codon. It might change the codon from a codon for one amino acid to another one. And so you introduce a different amino acid, and that might change. If you change the sequence of a protein, what might it affect? Everything, right? But specifically, the sequence of amino acids determines the protein shape. Shape is what I was looking for. So if you change an amino acid, you could change its shape, and then it might not be able to do what it normally does. And so now you have a defective protein. Also, that's the main one I want you to know, but obviously you can have a mutation that deletes a nucleotide, and that might mess up everything after it, because its codons are in 3s, so now you've shifted everything. It's called a frame shift mutation. You can also have an amino mutation that introduces a stop codon early on, so that actually stops protein synthesis. You only make that portion of the protein. So anyway, mutations can do a lot of things, but I want you to know that it can change the amino acid sequence, and that can change the protein shape and function. Boom! We just did all of that in like 15 minutes. So hopefully that doesn't sound too foreign, or hopefully it now starting to sound a little familiar, which would be good. Don't expect you guys to hear all of this and be like, oh yeah, that's a piece of cake. Maybe some of you, that's the case, but I expect most of you to be like, okay, that sounds familiar. It's something I can grasp if I study it. So now your job is to study this and to be familiar with the concept. Don't just memorize information. You'll learn it way better. So yeah, good luck. I think you guys are going to do great.